Okay, so thank you everyone for your attention, and I certainly want to thank McKinsey, Institute Scala, the conference organizers for the opportunity to speak here again. And my goal is to give you an overview of some of the issues related to transcranial direct current stimulation that my lab has been interested in investigating over the past 10 years, and where we see some of the challenges and opportunities in relation to mechanisms and optimization. And so I'll be talking both about modeling and animal work, so I'll be starting with the modeling. And so this is my disclosure. I actually will not be showing any clinical data today. So it's useful to start with a general overview of where tDCS stands today. And when you read many papers, certainly not all papers, but many papers on tDCS, they often start with two series of assumptions, one related to how the brain works, and then the other one related to how tDCS will change it. Many papers will describe brain function or dysfunction as a hyperexcitability or hypoexcitability. So for example, they will say that depression is associated with a low level of function in one part of the brain, or epilepsy is associated with too much function or hyperfunction in another part of the brain. And this will be extended to pain and so on. And this is not an idea that came out of tDCS research. This is something that is very broadly used in neuroscience and neurology. It has roots in TMS, in fMRI. And so that's where you get this notion of sliding scale, where you have a brain region and you're thinking about it as either being on one end or the other end. Not excitable enough or more excitable. And this is where tDCS comes in. So the notion is that with tDCS, you can move the brain along this sliding scale. So for example, this is usually a nodal stimulation is used to increase excitability, while cathodal stimulation is used to decrease excitability. So an example I just gave of depression, if let's say the prefrontal cortex is assumed to be not active enough, so the sliding scale one way, then you will apply a nodal stimulation to move the knob the other way. Or if you have epilepsy and it's focalized to one part of the cortex, you may be too far apart this way, and then you will use cathodal stimulation to move it down this way. And this idea of sliding scale and a nodal cathodal has been extended to many studies in tDCS to rationalize them, including for cognitive performance in healthy adults. And so an important question I think to think about is, does this make sense? What studies have we done as a community to support this idea? And is there a way where we can be more sophisticated, where we can approach both brain function and tDCS in a way that is more subtle and produces better results as a result of it? So we'll be starting with models. And this is an example of one. And I won't be going too much into the methodology, but certainly I'm happy to answer questions about that. All tDCS has at least one anode and one cathode, where the anode is the positive electrode that's connected to the battery, and the cathode is the negative electrode of the battery, to use that analogy. And current flows from the anode to the cathode. If there is a head in between those two electrodes, then current will flow across that head. And if there's a brain in that head, then the current will flow across the brain as well. And so the models predict where the current flows. So in this situation, this is a model, it's an MRI-derived model. In this case, it's of my head. There's a 5x5 electrode on the left side and a 5x5 cathode on the right side. The current is predicted to flow through the brain, as illustrated here. So you have blue is no current flow and green is some current flow. And you can see that the regions under the electrodes have the current flow. Now, it's certainly not exactly a square shape on the brain, but there's a general sense that the current flows where the electrodes are. We can take this a step further and we can ask, well, what about current flowing in or out? So under the anode, the positive electrode, the brain actually sees current arriving. So the current, the positive current, exits the red electrode, and it arrives in the brain, and so it flows into the brain. The opposite happens at the cathode. On the cathode, the current is being drawn out by the black electrode, and so it's flowing out of the brain. And so that idea is illustrated in the next picture here, where red shows inward current flow, blue shows outward current flow, and green shows no current flow. So I'll be using this color convention throughout my talk. So sometimes I'll be just talking about, is there current or is there not current? And sometimes I'll be talking about specifically inward versus outward current. And it turns out that this inward versus outward idea is considered very important. So as I go through my talk, I'll be talking about these 4 modeling studies. Some of them have a clinical component. And all the slides I'll post on the laboratory website, so these references, you'll have them available along with the data I'm about to show. So I wanted to start with this first paper, which was published now almost 5 years ago. And the bottom line with this study was that we modeled the current flow produced by conventional tDCS and compared that with a new montage that we called the 4x1 high definition montage. Now with conventional tDCS, you have in this case, two large 5x7 electrodes across the head. And when you do this, current is going to flow across the entire head. It's not going to just enter here and disappear, and then reappear here, right? So it has to flow all the way across. And so this idea is what you see in the model. You see the current is actually flowing all the way across the head. You'll also see in the cross sections that it does not stay in the gray matter. It's not strictly superficial. Just like if you were to put electrodes across a watermelon, you wouldn't expect to see the current flowing just across the top of the inside of the watermelon. You'd expect the whole watermelon to fill up with current. And that's what you're seeing here in this head model. And you can also see there's a lot of spread all over the head, in fact. So this montage, though it's been very successful, can produce, according to the models, some rather diffuse current flow. Now you can contrast that with the 4x1 montage. In this case, instead of 2 large electrodes, we have 5 small electrodes. And just without any quantification, just by eye, you can see that there is a pretty significant difference in the focality that you can achieve between those 2 approaches. Now to be clear, I've selected some rather extreme examples here to make the point. We can look at this 4x1 montage in a little bit more detail. The center electrode on the 4x1 can be either an anode, the positive, or it can be a cathode. If the center is an anode, then the outer 4 electrodes are cathodes. If the center is a cathode, then the outer 4 electrodes need to be an anode. And what happens is that, in this case, for example, with the center anode, it pushes in the current, and it is collected by these outer rings. And so the size of the ring, how wide we make this ring, determines how much of the brain we activate. And by making that ring smaller and smaller, we can focalize the current further and further. The other nice thing about this montage, according to the modeling predictions, is that it produces rather unique directional modulation. So often with tDCS, you have an anode and a cathode, and you're trying to balance out these issues of excitation and inhibition. Certainly Dr. Nietzsche showed many ways to deal with that. One of them is to use this montage, which produces largely unidirectional current flow. So if you just want to produce excitation, you would use a center anode. And so the main finding, which this was a purely modeling study, was that using high definition electrodes, you can improve the focality of tDCS. And specifically, the 4x1 montage seemed to be promising if you wanted to target a cortical region and target it with a specific focality. Now, it's important to emphasize that this does not mean that tDCS using large pads cannot be used in a way that's dose-specific. And certainly by manipulating the position of the pads, by changing their size, you could have very specific effects, not just on the global level, but at a gyri-specific level as well. So one study that we're working on now with Dr. Nietzsche's assistance is to go back to his foundational paper in 2000 and model current flow produced each one of these montages and specifically consider not the current flow throughout the whole head, but the current flow right through M1 and specifically the hand knob in M1, because that is the region that is likely most relevant for the outcome measure, which was TMS MEPs. So it's important to make that distinction. So moving on to the next paper, which was by Dr. Nietzsche as well, is to consider whether this new montage will have any effects on the brain. Especially when you start with a model, you need to be very skeptical. Dr. Nietzsche already presented this data in which, I think he presented an updated version of this slide, but it is the same qualitatively. What Dr. Nietzsche did is he compared the conventional montage, this is the M1 SO montage with large pads, with a 4x1 montage and a crossover across the same subjects. And just looking at the anode case here in the purple, you can see that conventional tDCS with a pad montage produces this typical initial excitation that then decays back to baseline. The 4x1 montage produced a very different profile. Immediately after stimulation, there was actually no significant effect when averaged across subjects. But over the course of the next 30 minutes, the magnitude of this modulation accelerated such that it was maximum at 30 minutes. And 2 hours later, this was still significant relative to no stimulation. And so this means that 10 minutes of stimulation with a 4x1 montage could potentially produce over 2 hours of modulation. Now, the TMS MEP marker is a gold standard in our field. It certainly has its limitations. For example, here, there was no change, but clearly something was not the same because stimulation had stopped and we saw an accumulation. But what this study showed by a lab that has been leading the field for some time is that with the 4x1 montage, current can be delivered to the region. And so a good question then is how much? And so this shows a side-by-side model. This was a more sophisticated model than the one I showed you earlier, but it's comparing the montage they used. There's a large pad here. You might be able to just make out the outline. There's a large return that was used here. And this is the 4x1 montage. And it's hard to make out the electrodes, but they're right. There's one small one here, and there's a ring around the outside. And so as I showed you before, you get a rather diffuse current flow, as you would predict using the pad montage, and a very small focus using the 4x1 montage. Now, in this case, the models are in both cases for 2 million. So it's the same total current being applied, being generated by the stimulator, but being applied to very different shaped electrodes. At first glance, it would appear that with the conventional montage, you're getting much higher peak current flow. And it's true. You see red here, and there's not much red here. But in this case, the region of interest was not necessarily the entire brain, but the motor region. And the 4x1 was put over the motor region. So if we look at that motor region in more detail, you can look at it here in this cross-section through M1, or you can sort of just, look right here with your eye. You can see that actually in M1, the difference in the electric field generated in the motor region is not as dramatic. Now with a 4x1 montage, much less current enters the brain than with a conventional. But the current goes through much less part of the brain, much less volume. So even though less current is entering the brain, the electric field you get may be comparable in the motor region. Another point which I mentioned earlier is you get a lot of deep current flow with almost all conventional tDCS montages. You can look at that in a specific sub-region basis, looking at the different deep brain structures. And I do think this is important because obviously the brain is a network. And so when you're using a conventional pad montage and current is flowing through deep brain regions, those deep brain regions may then be having a secondary effect, modulating the cortical regions that you're interested in, and may indeed be involved in some of the clinical effects that are observed. We could take this analysis one step further, and instead of looking at just where the current is flowing, look at directionality. So in this case, red represents current flowing in, and green would represent a region without current flow, and blue represents current flowing out. And now again, you can see that yes, the conventional montage produces much more diffuse current flow. But if you focus just on the motor region, and maybe even just on the hand knob, that difference now is not as dramatic. And so it's an important point. And so this paper, which is really foundational for high-definition tDCS, established that focal modulation with DC current can produce lasting plastic changes in brain function, in this case that lasted for a significant bit of time. Now certainly this is just the beginning, and there are several ongoing trials, clinical trials, human neurophysiology trials, looking at this technique, looking at dose response, and so on. But I am increasingly confident that the predictions being made by the models are relatively correct, as they've been borne out in all the tests that have been run. The next study I want to talk about was conducted by Dr. Edwards at the Berkeley Rehabilitation Institute, and we'll be hearing from him later in the afternoon. There's a bit of information in this slide, so I'll just unpack that piece by piece. One of the problems with tDCS is that it does not produce an overt response the way TMS does. So if someone's getting tDCS, you sometimes would ask yourself, is it working? Well, with TMS, you get a motor twitch, so it's very convincing. Well, it turns out you can produce a motor twitch with electrical stimulation as well. You just need to put in a lot more current. So whereas conventional tDCS uses about 1 milliamp, if you instead increase that to 1,000 milliamps, and now you use a very short duration pulse, you could produce an MEP, and this was established by Merton and Morton in the 80s, and indeed laid the foundations for a lot of the neuromodulation that was to come. But it is excruciatingly painful, which is why it's not very popular. What we did was, though, we used the 4x1 montage, and we applied high voltage shocks, so 1,000 volts. And we initially positioned the 4x1 montage over the motor region. And when we did this, we were able to elicit an MEP. So this is the 4x1 montage with 1,000 volts, and we're getting an MEP. Now we can take a conventional TMS coil, a figure-of-8 coil, put that over the same region, and confirm that that region is indeed the motor region, and we get another MEP. The next experiment we did is we took the 4x1 ring and we displaced it. And that's sort of what's illustrated in these pictures here. We can move the 4x1 ring either anterior or posterior. Now the model predicts that using the 4x1 montage, the current is restricted within the ring. So if the motor region is outside the ring, as in this situation here, there's a motor, and in this situation here, the model predicts that you will not be able to elicit a motor vocal potential. And that is what we found experimentally. Moving the ring anterior or posterior, the motor vocal potential went away. And we saw the same thing with TMS. Now in this paper, which is now published, we did a series of other experiments looking at incremental distances. We were not able to distinguish the vocality of the 4x1 from TMS for all the gradations that we tried and all the experiments that we did. What was very interesting is we did these experiments on 3 subjects. One of those subjects was myself and 2 of the other investigators. And we also conducted models of each individual subjects. We conducted MRIs from each individual subjects. And what we found was very compelling. And so that's these results here, which I'll explain slowly. We did 2 experiments. And let me start with the one on the right. In the one on the right, we applied the same dose to everyone. So everyone received 600 volts. I received a 600 volt shock. Dr. Edwards received a 600 volt shock. And we measured the MEP that would be produced for each one of us. Well, it turned out that in my case, 600 volts did not produce any MEP. It was not strong enough. Higher intensities could, but not 600 volts. But 600 volts could produce a very strong MEP in Dr. Edwards and in the other subject. Now, as I mentioned, we also had individual models from these subjects. When we modeled 600 volts on my head and compared that to the models of the other subjects, on my head, much less electric field was produced in the brain. So everyone is getting the same thing on the outside. Everyone is getting 600 volts on the outside. But because of my head shape, because of my head anatomy, much less current is being delivered to the motor region. Another way to do that experiment is to increase the current in each subject until you get a threshold MEP. So in this case, 1,000 volts was needed on my head to produce a threshold MEP, while in the other subject, significantly less. And once again, we modeled this on a subject-by-subject level, predicting how much current would be generated in the hand knob area. And the model was able to predict, with a reasonable amount of fidelity, how much current would be needed in each subject. And this was a factor of 2 difference, which is very striking. So there are 2 major takeaway points from this paper. The first one is it was able to validate the focality of the 4x1 montage, not using DC current, which is being done now in other labs, but by using high voltage shocks, which sort of produce a very unequivocal muscle twitch. But the other thing this study showed is that there was a lot of inter-subject variability. Now, as it turns out, this is actually rather well known. For example, in TMS, everyone knows that the percent output of the stimulator that you need to induce MEPs in different subjects can vary significantly. Well, electrical stimulation is no different. The difference is, though, that right now, in electrical stimulation, there is no individual dose correction. So when you run a study, everyone gets the same exact amount of current. And so there is motivation, based on what I've shown you, and effort, including data that Dr. Nietzsche has shown you, to normalize. And so with that in mind, I just wanted to touch on this last modeling study that we did that really reinforced this point. We modeled a wide range of subjects, ranging from super obese to borderline anorexic, with very different head sizes. And we asked several questions. One of them was, how much does the peak electric field in the brain change? Which is what I just showed you. The other one was, does the location of the peak move? So for example, if you use the M1 SO montage in one subject and the peak is in a certain region, will that peak be in the same place in another region? And then we evaluated some other issues related to tolerability and safety. And the bottom line is that people's heads are very different shaped, very different skull shape, very different level of fat. The brain morphology can vary significantly across subjects. And as a result of this, even when you apply the same outside dose to every subject, what's produced on the inside is not the same. And so I'll just, in the interest of time, jump to the main conclusion of this study. It reinforced what we found in the Edwards study of large differences across subjects, 2 to 3-fold just across healthy, typical adults. I'm not even talking about the presence of injury and so on. What's nice is that the models, we believe, can be used to predict these variations and make corrections for them. Now, we did model conventional montages and the 4x1 montage. The 4x1 montage actually has very high sensitivity as far as the peak. The advantage of the 4x1 montage is that the peak is always inside the ring. While when you use conventional tDCS, the peak is usually somewhere between the electrodes, not under the electrodes, and its position can vary pretty wildly across subjects. So these are these 4 modeling studies, and now I'd like to speak a little bit about the animal work that we've been conducting. I'm going to be speaking about work that we're doing at the tissue level. So this is using brain slices from animals. And again, I'm not going to talk too much about the methodological details, but I certainly can answer questions. And they're available, and these are the 4 publications that I will be summarizing briefly. This is the slide I showed you before that shows with a conventional tDCS montage, a lateralized montage, this notion that one part of the brain gets inward current flow and the other one gets outward current flow. And so the question we wanted to address with the animal studies is, we know this happens. We know that when you put electrodes on the head, you get regions of inward current flow and outward current flow. And we believe as a field that this inward current flow will lead to an increase in excitability, maybe an increase in plasticity, while outward current flow will lead to a decrease in excitability and maybe a decrease in plasticity or maybe a negative plasticity. And so the question is, how do we link these two? Why does inward current flow make the brain work, let's say, better? And so that's where we started. And before I go there, it's interesting to think about where we got this notion as a field. One source of this notion came from animal studies, going back to the 60s, even to the 50s, and Dr. Nietzsche already alluded to these. In these animal studies, they applied current to the exposed brain of rats, cats, and they recorded firing rate from neurons. And so this is what they saw. They would see a nodal stimulation causing an increase in firing, and a cathodal stimulation causing a decrease in firing. So this is pretty unequivocal data from animals. The limitations would be, one, they're using stimulation that is 100 times stronger than what we use in tDCS. And I think they needed to use that to produce measurable firing rate changes. The second part of it is, how do we explain that this makes you read better or gives you less pain, right? It seems like a very generic claim. How do we explain that? Let's say this makes you read less well, right? Or makes you more creative in tool use, right? All these claims that have come out. There is a bit of a reach here. The second seminal source of where this idea comes from is, as we just heard, from Dr. Nietzsche using TMS MEPs. And again, this is our gold standard data for dose control in the field. But it's important to note what he actually shows. He shows that when you apply tDCS, the response to a TMS pulse goes up and it goes down. And many people have cited his work to justify much more ambitious and abstracted goals. And so I would say there's a difference between what Michael Nietzsche says and what people says he says, right? And I think we need to be vigilant about that. The other sources of these ideas include imaging studies and clinical trials, and I think these are more often not conclusive than they are. And so this idea seems not as set in stone, perhaps, as sometimes assumed. So if we go back to this notion of anode and cathode and look specifically at what's going on at the cortex, when we have an anode, the assumption is that current is going to flow down, as I mentioned, into the brain. So this is the downward direction. And this is believed to produce a somatic depolarization and to hyperpolarize the other end of the cell. This is something that has been around now for several decades as far as how people believed direct current polarizes cells. And so a nodal stimulation would therefore be presumed to increase brain function because the soma is the thing that's important. And by depolarizing the soma, we are somehow, and this is a big somehow, making people better at math and so on. If you reverse the electrode, or we go to another part of the brain where the cathode is, the current is now flowing in the opposite direction. I keep pointing at this, but anyway, the current is flowing in the opposite direction. This produces an opposite polarization profile, where the soma is now hyperpolarized. And so people will then say, well, because the soma is hyperpolarized, perhaps, this represents a decrease in brain function. And so, if I were to fill in the blank here, one hypothesis is that cell polarization, and specifically somatic polarization of pyramidal neurons in cortex, provide a link between current flow and excitability changes, where soma depolarizing is what produces an increase in excitability. And so work we've done, going back about 10 years now, started to address this first question. So before we move on to this much more complicated issue, is this really true? Does direct current polarize cells in this way, and how much? So our first substantial effort in this direction was published in the Journal of Physiology in 2004. We used a broad range of techniques: optical imaging with voltage-sensitive dyes, neuron compartment model, intracellular recording. It's the nice thing about working with brain slices is that you can readily do all these things. The bottom line is that we were able to confirm this prediction of polarization and bimodal. So in this case, the current is flowing downward. You can think of this as cathodal stimulation if you're thinking about the cortex, but this is hippocampus. And you can see this bimodal polarization. For example, here, when we're conducting optical imaging with voltage-sensitive dyes, so this is a direct imaging of the transmembrane potential. You can see that the area, the somatic area, which is near the cathode, is being hyperpolarized, and the other end is being depolarized. And so the take-home message from this paper, which was supported by a significant amount of other work, is that DC stimulation really does produce this bimodal polarization. But it's important to emphasize that there is no such thing as a depolarizing field. You can say that nodal stimulation depolarizes the soma and hyperpolarizes the apical dendrite. That is correct. But to say that everything is depolarized is not supported by the data that we have and the data that other people have generated. The next question we wanted to ask is, well, how much? These cells are being polarized. For example, under nodal stimulation, the soma will be depolarized. Again, how much? So we're able to address this question using rat cortical brain slices. We're able to record from a large amount of cells from their soma as we apply electric fields and record how much these somas polarized. And this is what we found. We found that some cells didn't polarize at all. So it doesn't matter how much current we put in, the soma did not polarize measurably. The most sensitive cells polarized 0.3 millivolt per 1 volt per meter field. So the 1 volt per meter is the electric field. That is the unit that we use to describe the stimulation intensity locally around the neuron. And for a 1 volt per meter field, this cell only polarized 0.3 millivolts. Now the explanation for this morphological diversity is found in this paper and in other publications, and it relates to the shape of the cells. The more asymmetrical a cell is, the more the soma will polarize. Not the more the cell will polarize, but the more the soma will polarize, because where the soma happens to sit along the structure. So how does this relate to tDCS? So let me go back to this image. Now, the electric fields here flowing in the brain during conventional tDCS are no more than 0.4 volts per meter. So I'm not talking about the current we put on the outside, but the electric field that's generated at the gray matter. No more than 0.4 volts per meter, less than a volt per meter. Now the most sensitive cell is only polarizing 0.3 millivolt, there should be a millivolt here, per volt per meter. So if you multiply the electric field by the sensitivity to the electric field, what we call the coupling constant, you determine how much the soma will polarize 0.12 millivolt. Now 0.12 millivolt is a high estimate. It's the single most sensitive cell we recorded from. And assuming the absolute maximum electric field you would get in conventional tDCS at 2 milliamp with a montage that's lateralized. This is not very much. A conventional action potential is 100 millivolt. EPSPs that need to drive the cell to firing threshold would be significantly larger. So this is a very minuscule amount. It seems like it's almost less than the background level of noise. So it raises the question, fine, we have somatic depolarization, but it's tiny. And why would this tiny depolarization lead to a change in increasing excitability? It, by the way, won't lead to much of a change in firing. I mentioned before that the earlier animal studies used much larger fields, right? They were applying the equivalent of 100 milliamp. Well, there's a reason why they needed to. This won't produce much of a change in firing at all. And so where we were left with this second study, which was published in Brain Stimulation not too long ago, was that tDCS, we predicted, would produce cell polarization, but it would be a very small amount. And this is where we get the notion, other people as well, that tDCS is subthreshold, it's neuromodulatory. It will not make cells fire. It's too small. And so that raises the question of what does it do? And so we know that we have binodal polarization, as I just described, with anodal producing somatic depolarization, and we know that it's very small. So the next place we wanted to put our attention to was, well then how does this lead to a change in excitability? Why would something so small, below the level of noise potentially, make you smarter and help with your Parkinson's disease and so on, right? It's really a very important question to think about. And to address this question, I think it's helpful to go back to where we started, to Dr. Nietzsche's pioneering work, in which he demonstrated that tDCS can affect brain function, right? Which was something until then that we did not necessarily know for sure. The way he did it was by using TMS to a vocal pulse. And so let me try to draw an analogy for what that is and how we could reproduce that in a brain slice. You start by polarizing the brain, and then you apply a shock to that brain. And you see how that brain responds to that shock. And cartoon-wise, if you can think about it, here's our cell. The cell is exposed to an electric field going downward. We'll call this an anodal stimulation, leading to somatic depolarization. Now, this does not make the cell fire. Then, TMS comes along, produces a shock. That shock travels down the axons and produces an EPSP, an excitatory postsynaptic potential, a synaptic current, which the cell receives. Now, it may or may not fire. Now, we can reproduce this in an animal model with something called field EPSPs. You can also record intracellularly and quantify excitatory postsynaptic potentials. So I'm not talking about firing rate. It's very important to make the distinction here. I'm talking about synaptic strength, how strong things are connected to one another. So if I send the same volley here to an unpolarized cell, or a depolarized soma, or a hyperpolarizing soma, how much response will I see? And what we found was that for this particular pathway, a nodal stimulation, somatic depolarizing, made the EPSP bigger. Now the cell is not firing here, but the same synaptic input coming in here, which normally produced this size of a response, is now producing a bigger response. The cell is effectively, you can think of it as it's listening more closely. The same input produces a bigger response. This is an input relationship. And conversely, if you do cathodal, which would be blue here, hyperpolarizing soma, this response gets smaller. Now, in this paper, we looked at this in exhaustive detail. We considered different factors. For example, we considered pathways coming into the apical dendrite. Now, in this case, it's not immediately obvious what will happen, because the input is coming in here. So if I depolarize the soma, will this input become more effective or less effective? It turned out that across all the pathways that we tested and all the conditions, which were rather broad, we always found that this relationship held. We always found that when you depolarize the soma, the sensitivity to synaptic input increased. And again, this is sub-threshold, and we're not triggering firing. And so one way to think about this is that we have a dial here. And what tDCS is doing is not making the cell fire more, it's making it more receptive to input, or it's making it less receptive to input. And I think it's useful to show a very similar experiment, but in a different context. In this case, we're applying continuous stimulation, almost like RTMS, with a version of it in slice. And what we're doing is we're producing a train of EPSPs. You can see there's a response, 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 so on, and it keeps going. The neuron keeps responding as far as the synaptic input that it's receiving. It's not firing. At this point here, we've turned on the field, and we continue with this train. And what we found was that throughout the application of DC fields, this train was bigger. So as long as the anodal stimulation was present, the EPSPs that were arriving at the cell had a higher gain. They were listened to more intensely. Whether or not the cell was firing or not, the same input now arriving at the cell is amplified. And this was, again, was borne out through a range of tests and quantification, but essentially what we're showing in this publication is that when you are under DC and nodal stimulation, you can sustain a higher level of input. And the reason I think this is very translatably relevant is often tDCS is not applied on its own. It's often applied in conjunction with something. So tDCS in conjunction with reading, tDCS in conjunction with various forms of rehab. And what you're trying to do is you're trying to make that process more effective, right? You're trying to make the reading, right, accelerated. You're trying to make the rehab accelerated. And so you can very generally think of an analogy here where when you, for example, apply DC and nodal stimulation to the cortex, it's not that you're making it fire more. It's that you're making it for that period of time, and perhaps for a period of time afterward, more receptive to input. And it's a slightly different way of thinking about things because we're talking about soma depolarizing, leading to a change in synaptic processing, and enhanced synaptic processing, and inhibited synaptic processing. And I think there's a lot of mirrors to this in a lot of the clinical neurophysiology work that Dr. Nietzsche just presented, showing how we modulate synaptic function, we modulate what tDCS does. It's on potentially the input side, not the output side. So that was this publication here, from which we would just generally conclude that the most noticeable effect of weak DC stimulation is to change how synaptic input is processed. And this finding has been mimicked before us and after us in many labs that have shown this to be the case. So DC fields are very effective in modulating synaptic efficacy, whether or not the cells fire or not. Very briefly, I'll touch on the final study I wanted to talk about, where in this case we looked at gamma oscillations. So these were gamma oscillations that were induced in a brain slice by application of carbachol. This makes the slice oscillate at roughly gamma frequencies. Beyond that, this probably has very little to do with gamma oscillations in people, but it is a model of ongoing activity in a slice. What we also did in this study is we developed a detailed network model of the oscillation, and we modified this network model so that you could apply stimulation to it as well. So we had both an animal model and a network model. So this is not a current flow model, but a model of cells being coupled to each other and communicating and generating the same regenerative activity. And we applied direct current to this activity in a slice. And certainly there are many details in the paper itself, but one of the most significant findings we observed was that when you applied nodal stimulation, so this would be somatic depolarizing, you saw an increase in power. And when you applied a hyperpolarizing stimulation, you saw a decrease in power. And for short duration fields, this recovered. For long duration fields, I'm not showing this data, it does not recover. The sustained activity, the sustained, let's say, excitation persists after the end of stimulation for roughly the amount of time that you're stimulating. Now this is sort of interesting in itself, but it was really only the beginning of the story. One of the advantages of working with a brain slice, and furthermore having a computational model, is you can really ask at a very cellular level what is going on. Why is this modulation asymmetric? And another very interesting feature, so at the most basic point we can say, okay, we're back to the dial, right? And the dial is moving power up and moving power down of this oscillation. And so the state of the system has changed. But it's not quite so simple. You can see, for example, that when we first turned on the nodal stimulation, there was a very strong response. And then, even though the field was still on, the system accommodated. And we saw something very different when we did cathodial stimulation. There was a stronger inhibition initially, and then the system seemed to recover. And what was curious is that when we turned off the cathodial stimulation, so here the cathodial stimulation is off, we saw this very dramatic rebound effect, where the system seemed to overshoot and then come back. And we saw this both in the animal model, in the dish, and in the computational model. And we were able to dig into this in quite a bit of detail. And what we're able to find out, to make a very long story, let's say short, are two key features. Let me just start with those. One of them is that when you have an oscillating system, the system is extremely finely tuned. You have a lot of firing of both the excitatory neurons and the inhibitory neurons. They're very tightly connected, and you get this balance, a very tight balance, where if the excitation guides go a little bit too much, they activate the inhibitory interneurons, but then feed back on the excitatory neurons and shut them down. But if they shut them down too much, now the excitation affects the inhibitory less, you have less inhibitory feedback, right? And you go back the other way. And that's precisely what gives rise to these oscillations, this sort of little bit of resonance across this hyperexcitable activity, but the system is very highly wound up. And this explains why the system is so sensitive to electrical stimulation, because it is already so finely tuned that any small perturbation will lead to a very robust response. But it also turns out that because of this, the system can respond as well. So I showed you in the slide before, let me go back to it. You saw this accommodation. So you get this initial extremely high level of sensitivity. And then the system responds back. It's able to accommodate it. And I want to emphasize again, we don't assume this is a model of gamma in people or somehow related to binding or cognition. But what it does show is that when you have an active system, how it responds is a function of the state of the system. It's because this system is oscillating because it's oscillating at gamma, because it's behaving in this way, then it responds in this way. And we have another model of slow wave activity, and it responded completely differently. So the state of the system determines how it responds. And in this point here, not only is the power different, the entire state of the system is different. And that's what I tried to illustrate here. So in this case of a nodal stimulation, not only is the oscillation power higher, the entire state of the system, as can be measured by the efficacy of excitatory synapses and inhibitory synapses, has completely altered. It's not the same system. And so what's interesting to me is not that the power has changed, right? That's phenomenological, and that is interesting, and people have found that in humans. What's interesting to me is that this is no longer the same system. It's no longer the same gamma. And so I think it's reasonable to assume that the way this system here, if I can go back, the way this system here processes information will not be the same. So yes, its power is higher, sure, but what's more important is that when information now arrives, in this case at the hippocampus, that information may not be processed in the same way, and it may not be consolidated in the same way. So the processing of the system has changed. And this is similar, to conclude, this has actually a lot of analogies with the previous point. DC does modulate the power, but underneath that power, when you look at what's actually happening at the cellular level, what you see is that the synapses are no longer working as they were before. And the way information, incoming information, is now processed would be different. And I'm not going to get into this, but we also, in this case, looked at AC stimulation, and we were able to very robustly characterize the response of this oscillatory system to a wide range of AC stimulus. So if you work in tDCS, I would suggest you look at it. Let me just jump to a final slide, because I know I'm running out of time, and explain why I think this processing of input, a theory that we're starting to describe as sort of a gain control theory of tDCS, is very important. You can focalize tDCS, certainly with high definition tDCS, but only up to a point. And actually, the vast majority of tDCS is not done with particularly focalized stimulation at all. And so if I have a group of neurons here that's near the electrode, they're all going to be stimulated. I cannot stimulate one and not the other. They're overlapping in space, right? They could literally be microns apart. No montage we could ever design could stimulate these cells of interest and not these cells. But as I mentioned, a lot of tasks are done with combination with behavior, right? There'll be a behavioral intervention combined with the tDCS, which makes one part of this broader brain, one network, more active, right? This, and I'm actually showing this as outputs, but really this system here is now receiving more inputs. So if it's the network of the brain that's involved in reading, right? That network is now receiving more inputs as it's trying to process this reading information. And even in the case of the MEP experiments, you're probing that particular system. You're sending inputs to that particular hand knob to try to activate it. And so the broader hypothesis is that specificity can arise because only the co-activated network will be modulated. Because it's the only part of the brain that's receiving a particularly elevated amount of input. So for example, you're doing post-stroke rehabilitation, hand training, and that is generating a lot of input coming into a region, and you want that region to undergo plasticity, and now you apply direct current, and you can make that plasticity more effective in a very selective way. So this is a bit of a cartoon. We'd like to make this idea a little bit more specific, but I did want to point ahead at what we've been thinking about and the topic that we are now sort of most interested in. So beyond just making the current flow selective, making it functionally selective within a given region. And we believe that thinking about inputs instead of just outputs may be a way to do that. So thank you for your attention.
Well, Professor Bikson, and congratulations for exposure. Is there any functional study showing the response in brain function in patients submitted to tDCS and also TMS?
Good question. Sir, could you repeat the question one more time, please?
Okay. Is there any function study in patients submitted to treatment with tDCS and also TMS?
Oh, you're asking if there are any studies which have combined both
Yes,
TMS
combined.
and tDCS
Yes.
for the purpose of an intervention?
Yes.
That is something I think there might be people in the room with a lot more expertise than I'd answer with. I think the one study, there certainly have been. I mean, there's one study that even showed that there is a complex homeostatic interaction between them, right? Where if you do one and then the other, they interact in a very nonlinear way, certainly. There has been no study that I know of that combined focal high-definition tDCS as an intervention with TMS as an intervention, and that would be something very interesting to look at, because essentially you're focalizing using two different techniques on one region.
Thank you very much for nice work. It's a very naive question. You use 4x1 configuration for your tDCS, right? Why not 3x1 or, for that matter, 5x1 or even ring to electron configuration?
It's not a naive question, actually. It's the perfect engineering question. So 4x1 uses one center active electrode and 4 outer returners. Why 4 outer returners? Why not 2, 3, and 5 for that matter? The principle is that you want to emulate a circle of electrodes. So in the ideal situation, it would be 1 by infinity, and that's where we started with the montage. So 1 surrounded by 100 electrodes. But it is very burdensome to set up 100 electrodes in a perfect ring. And so the real question is, how few electrodes can you use so that the brain current flow still looks like what would happen if you used the ring? And it turns out that that's 4. So going from 4 to 5, you do not significantly change the brain current flow through the head. However, when you drop the 3, and certainly when you drop the 2, then for certain montages, it doesn't look the same. For example, a 1x2 may look more like a center anode with 2 little cathodes on the outside. Now that may be what you want, right? Because high definition can be deployed in different ways. So that may actually be a great, something you may want, but that's where the 4 comes from. It's a very good question.
Temos tempo para mais uma pergunta. Alguém?
Congratulations. We are talking about very small current when it gets inside the brain. Does it have any difference if you have a patient that is dehydrated, or if he gets 3 cups of water before the intervention in the electric bath or in the electric difficult?
So it's an interesting question. The question is if someone, let's say, was extremely hydrated or the opposite, dehydrated.
Yeah, very hot day, gets in the lab, dehydrated.
Would that change brain current flow? I don't know the answer. I would say that if the level of hydration or dehydration is significant enough to, let's say, cause a swelling or a shrinking of the brain, even by millimeter, that would have dramatic consequences on how the current flows. And the same thing would go for the ventricles. They play a very important role in shaping the current flow. that would certainly matter. If the salt content of the brain or the CSF were to change in either direction, that would also have a very profound effect on current flow. And so the composition of the CSF and the fact that it's so thin is so important to brain current flow that if that were to change, and I don't know if that could, it would affect significantly current flow.